This is a lab lecture for the other bacteria. Most of the tests that are done have been previously covered in other lectures, so there aren't very many to cover in this lecture. If you've listened to the uh, actual lectures for the bacteria before this one, then you'll know that a lot of these organisms are identified based on automated type methods now, or through some sort of serology type testing. Organisms that are anaerobic are typically obligate or strict anaerobes, and they will not grow in the presence of oxygen. You do have a few organisms that will grow in the presence of oxygen. They just may be a lesser growth, or it takes a little bit longer for them to grow. Anaerobes are killed within a few minutes if they're presented to oxygen, so the collection technique is very important if you're suspecting an anaerobe. So these cultures may be collected through some sort of like syringe or aspiration, and if it's something like a swab, then it would be placed in a special media that would prevent exposure to oxygen for that organism. If a anaerobic organism is suspected for an infection, then the presence of a foul odor or sulfur granules would give a good indication that it is an anaerobe causing the infection. And of course, if you're looking at the organism on an agar plate and it fluoresces, then that's an indicator that it is a anaerobic pathogen. So these organisms can be detected through a variety of methods, but if we're looking for direct testing methods, then we could be doing some sort of antigen type detection. And so this might be looking for something like Clostridium difficile, where you're looking for the particular antigen for that organism and some of the different automated methods. And then you could also see the organism with a gram stain. So with a gram stain, your anaerobes can stain poorly. So sometimes staining them in an anaerobic type environment, which may not always be feasible to do, can help this can help to improve the staining process and technique. Specimens are processed in a biological safety cabinet, and then these organisms are um, incubated in special conditions. So it could be something like an anaerobic jar, or some sort of holding jar, which you can see over here to the right, where a gas pack has been placed in there to remove the oxygen, and then it's sealed shut. And then that gas pack will remove the oxygen, and so that environment will stay anaerobic without the presence of oxygen. And you can do this same process with bags to put these cultures in. So depending on the lab and the amount of organisms or cultures that they get for anaerobes will depend on which method that they use. A lot of labs will just use the anaerobic bags. Once they're cultured, those plates are incubated with those anaerobic conditions for 35 to 37 degrees Celsius for 48 hours. After that time, they're taken out and they're examined for growth. If there's growth, it's worked up. If there's no growth, then it may be incubated for a little bit longer to try to detect those slow growers. And then it's also, those cultures will be inoculated into a thioglycate broth. And once it's put into that broth, you'll have two broths, and one will be capped very tight to keep that anaerobic environment, and one will be put on loosely so that it'll allow the presence of oxygen, so that if there are aerobic organisms as well as the anaerobic, then you have the ability to detect those organisms as well. There are several different types of media that are used for your anaerobic culture, so it can include blood agar. Your BBE agar is specific for your Bacteroides, so it helps to isolate that organism, as well as look for that colony blackening characteristic that it presents with. And some of your other augurs that might be used would be to isolate specific organisms, or to enhance their growth, or to inhibit other potential organisms that could overgrow that culture plate or overgrow those anaerobes. Your CCFA augur is used for Clostridium difficile, and so it would be selective for that organism, and you would be looking for that yellow colony appearance of that organism. There are some commercial systems available for anaerobic culture media, and so something like this would be where you actually get the culture plates pre-packaged. So they're actually produced, packaged, and shipped and stored under anaerobic conditions. So they're sealed in those little pouches to keep that agar plate in an anaerobic environment. And then once you're ready to set up that culture, you would actually take those plates out of that packaging, set it up, and then return it back to an anaerobic environment. With examining anaerobes, you're typically going to find a mixed culture because you do have so many normal flora, anaerobic flora. So that's why trying to get that differential or selective type media in that culture setup is crucial, so that you can eliminate some of the other anaerobes that may be present. And it's possible to have a couple of different anaerobes that would be the culprit of the infection. And so once you're looking at the plate and you're looking for the organisms, and you determine which ones you feel like are the pathogens or causing the infection, then you would isolate those out and do a subculture on a separate plate and regrow those organisms. And then of course you have the backup broth that you can do a gram stain on or subculture if you need it. Since these organisms are very sensitive, you could lose them through the subcultures. And of course, if you culture those plates and you don't see anything after 48 hours or several days later, then you could do a gram stain on that broth to see if there were organisms that grew in the broth. And of course, you've also got your automated methods like PCR testing to detect these organisms as well. When a specimen is submitted for possible anaerobic culture, or if the physician suspects an anaerobe and sends an anaerobic culture, then the sample may be reviewed with a gram stain for a direct testing method. So for example, if it was a aspirate fluid, then the gram stain would be made of the fluid and reviewed to see if there is any sort of organisms present. And so that would help give a preliminary report. You still wouldn't know whether or not it was a aerobic or anaerobic at this point, but if you did see spores, that could kind of lead you in one direction or the other as well, and it can kind of help correlate with what you could potentially be seeing in that specimen. So you would also set the cultures up, so you'd treat for isolation, and you would set it up on the anaerobic media. Each facility kind of picks which particular media they want to use, so they kind of know which organisms are more common in their area, so they may base that on what their area presents with. And then after you've set that culture up and you've let it incubate, usually 48 hours, then you would examine those anaerobic cultures. But typically when you set up an anaerobic, you're also going to do an aerobic, so those would be reviewed the next day. And then once you determine you have growth on your anaerobic plates, then you would do a gram stain, or you would subculture to something like a blood agar or chocolate, depending on what you're looking for. And then with your gram-negative organisms, you would also add those antibiotic disc to the plate so that you could look for susceptibility or resistance to those, and you may also add a nitrate disc. There are a couple of different methods for the nitrate testing, but a disc method is one of those methods. To identify these organisms, some of them can be identified based on presumptive identification using some of your spot testing, like your catalase, spot indole, as well as the antibiotic testing, lipase and nitrate reduction. And then a definitive identification can be done after using other techniques, including commercial identification systems or biochemical kits. Here we have the vancomycin, kanamycin, and colistin test. So with this test, you would actually set it up like you do your other susceptibility testing, where you plate your organism over the entire agar. So you would create a lung of the bacteria, and then from there you would actually place the antibiotic disc onto the plate. And then once you place those discs, you would incubate that agar plate, and then you're looking for growth around those antibiotic discs. So here is some examples of those patterns. So this very first column, you have your vancomycin, your second column is kanamycin, and your colistin is the third one. And so with each of these different rows here, with our first one, you've got growth all the way up to that disc, so this would be considered resistant. And so that's the same for all of these. They're resistant. This second row here, you've got sensitive, because you have growth up to a certain point and then it stops growing. With niacin tests, we're looking for the accumulation of niacin in an egg-based medium, and the result of this results from the lack of an enzyme that's able to convert the niacin to another metabolite in the coenzyme pathway. And so this is characteristics of Mycobacterium tuberculosis, and there's also a few other organisms that will present like this. But niacin is measured by a colored end product. And so what you're looking for is a yellow color change, and you can see that in the image here, where A has a color change, and that is considered positive, a little sideways there, and then B would be considered negative. Nitrate reduction test, again, there are a couple of different methods. You can have a disc method as well as broth methods. And this is looking for the presence of nitrite, which is detected by red products or a pinkish type color change. So if the enzyme has been able to reduce nitrate to pass nitrite to gas, then you would add the zinc dust, which will convert the nitrate to nitrite, and that will detect the lack of nitrate in the reaction medium. So positive would be the development of the pink color, which indicates the presence of nitrite, demonstrate the organism's ability to reduce nitrate to nitrite, so you have that pink color there in the center. And then a negative would be no color change. The organism may have reduced nitrate beyond nitrite, and so what you would do is add a small amount of the powdered zinc to the negative tube, and you're looking for the development of a red color, which indicates that there's unreduced nitrate that was present in the tube, and the organism would be nitroreductase negative. And you can see a negative reaction here. This would be your control tube.